Right. Next speaker, our guest speaker, he's a Professor Tatsuo Watanabe. He's a research professor at the Gene Research Center, Graduate School of Life and Environmental Science, University of Tsukuba, Japan. Can you hear me all right? His research of interest are molecular genetics and cytogenetic study, the production of transgenic plants, and biosafety and bioethics. Sustainable germplasm on polysomic polyploid species. The topic that he will talk today is breeding cases on polyploid species. Welcome, Dr. Watanabe.
Thank you very much for introduction, and nice to see you all audience, and thank you very much for the organizer giving me this opportunity. So let me jump on my subject, and I have the 6 study coverage of topics, but mainly I will talk on number 2, potato as example on polyploid crop breeding. You see a lot of crops, and talking about how many of plant species you can use for eating, biting, chewing, medicinal use, you may count 80,000 species on the planet yet. But how many we are handling, dealing for the crop improvement? Very little proportion. And indeed, talking about the plant kingdom, that already Dr. Hans de Jong mentioned that many species are classified into polyploid origin, or the high genome size, or the real the polyploid in terms of the genetic type of behavior. And already a lot of classification and were made on the polyploid topics in 1970s to 1980s. So you go over all the literature, classical literature, you can find out a lot of supporting documents on polyploid on crop species. So especially root and tuber crops, because they can make a vegetative propagation, they can maintain heterozygosity, and also the possible to stay as a polyploid. So not only potatoes, sweet potatoes, and some of taro, yam. Cassava usually diploid, but there is effort to make artificial triploid and tetraploid in breeding history. And you talk about the locally important the tuber crop. You go to Andes. You may look at the oca, wiko, mashua. Those are also the root crops, and they could be the polyploid. Like the oca, oxalis is typically with a polyploid tree. So my major interest actually on root and tuber crops. And in Asia, I'm looking at the gingibraceae. Actually, I would like to share some of our research on occurrence of triploidy and some of metabolite contact with the triploid. And also some of underexploited crop like this. Can you tell me what is this on this floor? This one, arrowhead, common in Asia, the East Asia to India. So one of underutilized crop, Sagittarius species, which stay in old continent. Also, you can find in new continent in United States. So there are a lot of the species which could be maintained to use as a crop, and especially also with the polyploid possibility. Okay, so finish introduction, and let me move to potato. So the potato, as you see, the original the places are around the Andes. And you go there, you see that those are the diversity of potato crop. And these are not only tetraploid. Could be diploid, some triploid, and the majority tetraploid, and a small occurrence of pentaploid, five-ploid. And the potato is typically you may call autotetraploid or the tetrasomic tetraploid in terms of genetic behavior. And also, the complication of potato is, as Dr. Hans explained, there is the unique the chromosome behavior in meiosis. Depends on interest of locus, stay distant or close to centromere, segregation change. So you may call chromosome or chromatin segregation. So that Dr. Hans explained. And the tetraploid, but you can see also the hexaploid, pentaploid, triploid, different the ploidy in species. And the actually potato, the tuber-bearing solanum species consists of diploid 2 hexaploid. And among them, so-called polyploid species can have a multivalent formation, but not always. A case of cultivated potatoes, so chromosome number is 48. So basic chromosome number is 12. But it doesn't mean all the 4 sets of the 12 sets of chromosome form the quadrivalent. Actually, quadrivalent occurrence is the lower than 8 quadrivalent per the genotype. So the chromosomal behavior is the complicated case in the potato meiosis. And also, so far potatoes, cultivated potatoes are tetraploid. You can see diploid wild species as source of potato crop improvement. There are about the 100 some diploid wild species which contain different sort of disease. Insect resistance could be the abiotic stress tolerance, like the relatively high salinity tolerance. And also, common potatoes are tetraploid. You can induce haploid artificially. There are quite good inventions made by 2 groups. One is Wageningen, no time ago, by Dr. Hansen, found a diploid which used to be a pollinator to cross with the tetraploid, and some chance that a pollinator introduced the pseudogametic parthenogenesis I show you here to make the haploid. So some people may call a dihaploid because of ploidy, but anyway, haploid induction is possible besides you do anticulture. But actually, more useful the haploid were produced from this pseudogametic parthenogenesis in potato genetics and breeding study. And potato genome was reported in 2011. At that time, the report was made based on diploid potato hybrid, and actually part of it made from the haploid. And also polyploidization, diploid, haploid at ploidy level of the 2x can be put back to tetraploid by use of the 2N gametes. And mostly 2N gametes, you may call unreduced gametes. What is the difference between 2N gametes and unreduced gametes? So unreduced gametes means the two means. One is the chromosome number not reduced, and also genetically unreduced. So the combination might not occur during process of unreduced gamete formation. But the 2N gamete is not only on the unreduced number of chromosomes, but also could contain some genetic change during the event of the meiosis. And also the diploid to hexaploid, even with the same ploidy of tetraploid cultivated potato and the tetraploid wild species, there is a hybridization barrier, principally controlled by the gene factor called endosperm balance number. So the endosperm balance number, is determined by a couple of genic components. And this one can be predicted by looking at the genotype or a specific species. Then you can predict the success or failure of hybridization between cultivated potatoes under the specific wild species. So these are the one slide set, but the story could be over 50 years of effort by many scientists. And I have the brief summary of your comparison of Arabidopsis and the potato in terms of some of the basic reproductive character and the genetic component. Anyway, potato is typically heterozygous, also besides the tetraploid, and also potato option of reproduction is a vegetative reproduction by tubers or by cutting. So you perpetuate the same genotype by vegetative propagation. But many of potato, cultivated potato can make flower and can be fertile, and even you can make a self-pollination, and you can make a botanical seed. So when we talk in potato industry, potato seed means the planting material, that means the tuber. But we can also make the two botanical seed by flowering and also the crossing or selling. So those are also the characteristics of potato when we talk about the breeding, the best line. So reproductive biology I mentioned mostly, and but the reality of potato breeding, many of modern cultivated potatoes, tetraploids, have a problem over the flowering characters. Some do not flower under the given photoperiod condition. So flowering associated with the long day condition or short day condition, usually moving to short day condition, also flower induce also tuber. So the controlling flowering is one aspect of potato breeding. So usually you don't close the potato in cultivation season by artificially controlling lighting conditions to induce the flowering, then the process. So when talking about the baseline of plant breeding to induce the useful genetic variation, the cross hybridization between cultivated potatoes were the biggest option. But by looking at the resource available, there are many wild species also existing, and also genetic stock already collected and staying with us. So how about utilizing those the wild species? Then they need to look at other characteristics like the crossing compatibility occur or self-incompatibility may occur, and also the sterility occur, male sterility, female sterility. So there are a lot of need to validate the reproductive character of the types of the interest before starting those of potato breeding by the sexual hybridization. And by looking at the modern potato varieties pedigree, many are closely related. Even the European potatoes have the bloodline of American has a bloodline of European potato. So the widening the genetic base was the subject in 20th century, and actually a lot of effort done like in the European potato breeding program. And going back to 50, 60 years ago, it was the beginning of considering utilizing the wild potato species, like a diploid species and some tetraploid species. Because of same chromosome number, it was thought that easy to use tetraploid wild species. But the reality was not so simple, but I'll tell you later. And talking about the potato genetic difference, the potato wild relative. Now the tomato also, the genus name is the Solanum. And actually genus Solanum is very big. And except the eggplant you have in Asia, the Solanum is in the New World. But the case of the wild potatoes, you have, I've already mentioned, diploid to hexaploid. And the diploid is principally self-incompatible. And the tetraploid, the hexaploid, self-compatible. Even tetraploid species and the hexaploid species, they are disomic polyploid. not like a potato. Potatoes are the polysomic, tetrasomic, the tetraploid, but some of wild species are disomic genetic behavior. So when you cross the cultivated potato with the disomic, the wild the species, disomic polyploid wild species, there is a mitotic complication that you may have a failure over hybrid. So need to know what is the cross compatibility and the result in the hybrid. So also looking at the evolution and how ethnobotanically the local people use. Although we have cultivated potato mainly as a tetraploid, but going to the Andean region, you can pick up a diploid cultivated potato, the good one like a Solanum pleja. Actually, that one is a high quality eating group that also is used in modern potatoes that you see in European market or the Japanese market. And talking about evolution, we may talk about the different sessions. Those are the wild potatoes are highly associated with the integration with cultivated potatoes. People potatoes, and after tetraploid selected, constantly wild potato has influence to tetraploid potato Andes to have a farmer that participates in plant breeding. So breeding. So the potatoes are the tetraploid, one over concerns even single gene inheritance character. There is more complexity of segregation. I'd like to overview again after the explanation of Dr. Hans. And many of the combined character has high genotype by environmental interaction. So that means that you need to evaluate one genotype at a different location to see performance blocks. And those are the classical quantitative genetic statements, but many characters are additively influenced. That you need to look at like a yield component. And also heterozygosity is the important part to maintain productivity of plants. So how to maintain heterozygosity with additive characters? So those may be characteristics were the challenge the factor for potential. And the modern science give you a lot of the transcriptomic analysis, and you will get the idea of how you can have each locus gene interactions. Now we can say epistasis is also another aspect that influence potato performance. And generally speaking, potato breeding, because of tetraploid, more segregation than diploid, and often the multiple torreya the subject for breeding. So need to have larger the population to select. But you don't need to have an integration from F1 to F2 and selfing. Once you have one hybrid the progeny generation to select, you select some candidate, you pick up, you maintain other potatoes, vegetative reproduction to finalize some candidate, right? But yet, with the orthodox approach of potato breeding, you may need 10 to 15 years. And also, it's not like a seed crop. You need to propagate the potatoes to plant. And the one potato plant may produce 10 planting materials, plant 10 seed potatoes. So, but if you have one rice plant, how many seeds you can have? Maybe 200 or so. So the propagation of planting materials are slow. So the breeding program usually identify candidate clone, they start propagating seed potatoes. So otherwise they cannot catch up with covering enough over the acreage to grow crops. So the seed program is in parallel, equally to other crops, are very important to start the production. So those are background. And anyway, this one is the simple picture. It's not a tetraploid by tetraploid crossbreeding. This one is the one of the scheme in parallel European and North American. They worked on around since 1980s to employ a diploid the species, and also the diploid has the simpler genetics. So you can work on diploid potatoes, make a haploid from a cultivated tetraploid, and also the use wild species to cross with haploid, or use cultivated diploid species to select or cross with those of haploid wild species haploid hybrid. You select at the diploid. So simple genetics, and you can combine different target characters. So that one is starting over utilize the diploid and also work on diploid genetics. And how to make those diploid to tetraploid? You may go simple crossing, or you afterward I will talk about twin gametes. Natural occurrence of twin gametes, especially twin pollen, is common in tuber bearing solanum. Then you select also for twin pollen at the diploid and cross those are the twin pollen forming the diploid genotype to tetraploid. So those are the recent scheme utilized in the past maybe 40 years for the introducing the wild genes, wild species characteristics into the tetraploid cultivation. So this one is one scheme set up in potato, and but also this scheme modified at different crops. Could be in blueberry, strawberry, or the sweet potato that I'd like to share some of them. So potato scheme is not only potato standalone. So modified scheme, the work on diploid and the polyploidize by using the principally twin gamete and make the tetraploid hexaploid. Polysomic polyploid genetics, Dr. Hans talked. So I wouldn't touch much, but I just run over the some confirmation. So I have a comparison of diploid, tetraploid, hexaploid, because I'm going to talk about the sweet potatoes. Sweet potatoes are hexaploid, and the basic chromosome number 15, and the hexaploid means 9. And then you assume that the one locus, two allele, large A, small a, and the diploid, you have a gametic output of large A or small a of 1 to 1. And the tetraploid, like potatoes, if you have a balanced dialectic condition, or you may call it duplex, and then you can have a clique group of gametes. So large A, small a, and a small a, 1 for 1. Dr. Hans indicated 2, 8, 2, but the same ratio. So you have the more allelic gametic combination going to higher ploid. You go to hexaploid. So balance the dialectic condition at the hexaploid, and you can have a 4 class of gametic output, and the ratio is 1991. So you imagine this gametic genotypic output combining to make another gamete to next generation of zygote. The segregation in the progeny is bigger with the higher ploid. This one is just theoretical, but the reality is so. And the moving from gamete to making progeny the segregation observation, like a diploid, I say Arabidopsis, but any diploid, you make a heterozygote cross a progeny, the genotype 1-to-1 segregation. If a large A is the dominant, you have a 3-to-1 phenotypic balance. And tetraploid, you go with the duplex, the balance, dialectic, the condition, progeny genotype goes 1, 8, 18, 8 to 1. So you compare with the heterozygote crossing at the diploid. Tetraploid has a very the wide segregation pattern. And taking large A as the complete dominance, you have a phenotypic segregation over 35.1. If you want to have a recessive to select, your chance is the 1 out over 36. So you need to have a larger progeny to select if you want to have this. Or if the additive effect take place, all large A are equally effective to have the some like the color or the yield component. You want to have this one. Again, chance is 1 out of 36. Then you go to sweet potato. We go later more about sweet potato, but let me use this slide set, and please remember this. Balanced dialectic, 2 allele, the 4 type of gamete combination. You go with the, please look, I don't count 1. The peak is 164 going down to 1. So if you want to have either extreme of all small a or large A, you have 1 out of 400. So if you want to select either of extreme, you need to have large project. Even predicting by this kind of segregation, you have a lot of trouble to prepare project population. The case of potatoes, actually fine. You pollinate the potato and you get the berry, like a small green berry, like a cherry tomato. You know how many seeds you may get from this? Any idea? Like same as the tomato, you can get 200 to 300 seeds per cross. So having enough amount of hybrid seed is possible in case of potato. So you can manipulate that. Although you need to handle 20,000, maybe 100,000 seedling to select, you still can generate enough amount of seed. So it's possible in case of potato. But sweet potato, I'll talk about it later. Although you need to have large population, actually reality of sweet potato is not so big. So if you have idea of morning glory as a relative of sweet potato, how many seed morning glory can produce from one capsule? Maximum 4. And I will talk later. Case of a sweet potato crossing, there are some reproductive difficulties. So not 4 seed per capsule. So that's how the people cope with the orthodox plant breeding at for example, potatoes and also hexagon sweet potato. And the orthodox genetics view, because one locus can have the 4 different alleles at the tetraploid. The case of hexaploid, 6 different alleles as a possibility. And also, you consider additiveness, the 4 allele additive, 6 allele additive, or the different allele interaction. So there are different possibility of gene matching. Here I gave the 4 allele possibility, and at the tetraploid, those 4 allele can have the interaction. Like you talk about biochemistry, there could be different biochemical products. And already multivalent issue is explained by Dr. Hans. Depends on the position of locus relation to centromere. There could be a chromosome segregation. There could be chromatin segregation. So even when you have a 2 allele condition, usually like the large small a, already you have seen in the previous presentation by Dr. Hans, you wouldn't see large the genotype, the gamete, but the chromatin segregation takes place with 1/6 chance you could have this rare allelic combination. And you may ask me, is that true, proven by some testing? Yes, those are tested by with the help of the molecular marker studies. It's possible. So this one is one of a background scientific complication over the polysomic, the polyploid. This case is the tetraploid potato. That you may need to keep in your mind some of the unexpected combination compared with diploid food. But anyway, this one is a summary made on the case of potato genetics. And now we have the NGS particularly available by looking at the dose of variation. And also the talking about the tetraploid potatoes, because you can maintain as the vegetative propagation, you may maintain built-in heterozygosity by keeping it as tuber. So the germ plasm enhancement, some of the practical examples that I was involved in the past using potato wild species to take that character resistance trait into the potato tree. And also the potato often mentioned two quantitative trait such as the insectoid tolerance consisting of different gene components that not only looking at heterozygosity per se, the specific genetic component that stay in chromosome 1, chromosome 5, maybe chromosome 12, those should be transcripted altogether without a segregation. So including those of topics of heterozygosity and the quantitative trait, there was the enhanced use of diploid breeding combination with the gamete. Why? First, let me go with a simple example of how possible to make the potato breeding. This case is a virus resistance potato virus Y commonly occur in the world. And the case of tomatoes, often you don't see so-called extreme resistance. Case of potatoes, we have like a potato type, the confined, the virus, this is confined with the dead cell. With that, we can avoid the spread multiplication and the spread of virus. So the potato virus Y resistance is one example. Actually, you can say that the transfer is easy. But there are different sources of resistance that need to identify. That's how we worked on this virus resistance called RY antigen. The case of the potatoes, there is a designation of genes made by the potato research. So, and this one is a bit jumping to complication of potato genetics at the tetraploid by incorporating some the genetic marker already done 20-some years ago. So genetic marker, like this case combination of RFP and also the modern, I don't say the modern, but the newer markers, you can have the potato resistance breeding easier. So this one is RY antigen genes identified on chromosome 11 of potatoes, and this marker simply simplifies by the scar markers, and even now that scar marker used at many places. And this one is the RY antigen gene baseline was an ADG2 fragment. It was studied in depth about the structure of a resistance gene. And this one is a typical resistance gene like the structure having some of the domain. And the RY antigen gene study also helped a lot of finding out a different type of potato resistance gene, not on potato virus. Why? So that's why I'm mentioning about the orthodox part of study done, especially by my colleagues at Finland. They did this marker study. And some domain like a kinase region common, and by looking at this region, the marker developed, and these marker help for selection of RY antigen. So although this one is single dominant gene selection, you talk about the seedling selection, and you don't need to wait for multiple virus the infection testing. It helps a lot for the base character built in for potato breeding. And this marker work high chance with many the genetic background in European continent and the North American continent. That's how although the work done 22 years ago, this marker set used yet that. And also the uniqueness is that the RY antigen is one source from the cultivated potato relative indigenous. But you look at the availability of source of resistance in wild species or cultivated species, you could find that those different genes. But genes don't stay in chromosome level. Could be a different chromosome, like a chromosome 12. So studying those multiple occurrence of resistance gene to PBY. So pathologists, biologists find the common feature of the virus resistance. So it's not only direction toward the application of potato breeding, but the baseline with study of availability of resistance also help to have how is the occurrence of resistance, evolution of resistance gene in potato map done a group of Germany and the US, and that was a baseline for potato genome published in 2011. So this one help a lot for the present the tetraploid potato study. And also the talking about Wageningen, they developed the AFP marker 20-some years ago. And this one also helped to work on tetraploid potatoes. Before that, it was not easy, even with a diploid potato genetic map. So there are a lot of technical incorporation to help the potato genetics. And the question goes to I mentioned about 2N gametes, 2N parent, and how the potato become a tetraploid. And Dr. Hans also explained the two possibilities of mitotic polyploidization and meiotic polyploidization. And the case of potatoes likely with a meiotic polyploidization by function of 2N gametes. And the question made going back to some of all the work, including myself, the genetic consequence and the significance of polyploidization by somatic doubling, mitotic or sexual, how to be advantageous in the evolution. That is the more theoretical part. But by looking at the occurrence of 2N gametes in plant kingdom, especially at the angiosperm, many cases are reported, not only tuber-bearing surrounding. So that's how likelihood of the polyploidization in especially angiosperm would have a higher association with sexual polyploidization. Just a background. So, but how easy to work with potatoes finding out 2N gametes, especially 2N pollen? Just use the acetocarmine glycerol stain, and find out relatively larger pollen. So the diameter is 1.2 to 1.3 times larger. By volume, it is about twice. And the occurrence in terms of frequency of sustainable pollen is not very high in large pollen. But you have two ways. Selecting for relatively large pollen production, relatively low temperature flowering condition, or once collect a potato pollen, shave a small one and keep a large one, and used for pollination. And the advantage of potato is that it's like rice. You have to pollinate daily, every day, take a fresh anther and fresh pollen. But a potato pollen, you take a potato anther and the dry and shed the pollen out from the anther, and you can keep the 1 month in refrigerator, and the case of freezer, probably maximum 6 months. So with that, you collect the pollen, and when the mother plant ready, you pollinate with the selected large pollen, pollen. So practicality to work on potato, so system set up to work with diploid, with potato. And the quantitative genetics is one question, and the heterozygous is another that I mentioned. And also this one, Dr. Hans already explained. There is a major two meiotic mechanism explanation over two gamete formation. So the FDR, first division distribution genetically, and the second division distribution genetically, how the transmission over the heterozygosity made. So I have an indication in relationship with the centromere, crossing over, and the pair the loci located maybe here or here. So it depends on where the those over the heterozygosity or locus of interest state, transmission rate over that the heterozygosity is different. So the case of FDR, so the centromere to first crossing over position, 100% heterozygosity can be done. And crossing over to the end, distal end, maybe 50%. But talking about SDR, centromere to crossing over, heterozygosity loss. But the crossing over to the end, heterozygosity can be maintained. So by knowing this rough sketch of how heterozygosity can be transmitted, and by looking at the overall the gametic, the genetic construct, you can think about how you can transmit the heterozygosity and the quantitative the correct together to the project. So using this the baseline studied a lot by different researchers. I have my own prediction made, and I don't read over. And I made the possibility over the combination of the Tolle, the characteristic, Tolle allelic interaction, the conservation can be transmitted. And I have a number of locus, loci, and when FDR is used, when SDR is used, when crossing over, P means the crossing over occur, how much the transmission over allelic interaction could take place, you can predict out from this kind of simple figures. So the potato breeding or any breeding, if coming with a dose of genetics the explanation, it is much easier to determine the population size or segregated project. And how it's solved. So this one is a granular trichome example done at the Cornell University, and myself, I was involved in this character. So granular trichome, tomatoes also have a trichome, and a group of potatoes has the wild species which has a granular trichome control the insect. This case is the red spider mite, and there are typically two groups of granular trichome. So the type A is the short and the large droplet, and the type B is the longer the hair with the small droplet. Both commonly have a character to have the smaller molecule, like a sucrose ester or phenol, and by those of smaller the insect or the mite touch on that, this droplet break, oxidize, and inside a small molecule of the polymerize and become glue. So those are the insect flap on the surface of it. So it is a natural protection mechanism by wild species. But looking at the classical approach on QTL analysis, So those are the characters are controlled by several loci. So how you can transmit this combination from parents to progeny. Usually you can have a large segregation. So working on tetraploid, you have a chance of a big segregation. But you use a 2N gametes, maintain, especially use the FDR 2N gametes, you may transmit a lot of a combination together from diploid to tetraploid. So that's how the initial work actually done at the diploid, and diploid the germplasm line made and crossed to tetraploid, and consequently those characters transmitted tetraploid potatoes. And I updated the slide set, but this slide set doesn't have a resulting tetraploid potato varieties. It was made by Cornell University, Dr. Bob Tracy, and the varieties were used in Canada, Prince Edward Island, called hairy prince. So it's possible to make the complicated trait transmitted to progeny and variety. And I have more the explanation of trichome trait transmission, tetraploid potatoes with trichome trait genotype cross, and how much genotype progeny could have a trichome. So this one is the selection, the result of example. So it's possible to transfer complicated quantitative trait from diploid to tetraploid and to select for target character. So anyway, the summary to here, the polysomic polyploid have a uniqueness in terms of they maintain genetic property. And but the question is the large segregation and the combination of a constant trait. That you can cope with that if you have the genetic base of information and the genetic resource. And the potato as an example, but then let me move to other crops. And I have my the references on the present the achievement, especially on you may have the genomics question. I have some recommended reading here that you can easily to get from open journal. And yeah, 12 gamete topics is relatively old, but you can go back to all the references that the fact I mentioned is true to confirm. And myself, it's not the simple work. So I have several reviews that I made, and this one also the open journal that you can confirm how is background story of work done. Okay, sweet potatoes. I've already mentioned that a large segregation occurs even with two alleles. How you cope with it? And sweet potato may not be important in Thailand. You have the starch crop more cassava. But talking about the global sweet potato production, it is mainly Asia and China. But also the increasing the demands coming out, for example, in Indonesia. So it's not a negligible in terms of the capacity as a crop, even in Thailand. So I mentioned about the hexaploidy number of chromosomes is the 90, and some background information I built in here. But the one of the problem of sweet potato is the cross incompatibility in cultivated hexaploid sweet potato. So cultivar A cannot be crossed with cultivar B because of cross incompatibility genetic system. So although the hexaploid, the crop, the variety genetic bases are narrow, this the cross incompatibility scheme also more narrows down possibility of cross. And also the sweet potato, although you have the big segregation to produce progeny, sweet potato is the morning glory relative to make a capsule to make seed cell. And the one capsule can have 4 seed maximum, but average seed set, even when you cross very well, 1.2 C per capsule. So you need to make cross to have a lot of seed. Even the smaller population size to start with, having several thousand of seed would take a lot of time. So that's tedious. That's how the breeding is relatively slow in progress. And the more problem is many varieties do not flower very well. So you have to have a conditioning. So you need to graft sweet potato scion to the morning glory relative to induce flowering. So the artificial flowering possible and the seed set possible, but you need to set up. So that's tedious and important. So that is another topic. And the other way around, you look at the possibility of source of useful genetic variation. You may pick up the wild species. Yes, wild species occur, especially in northern part of South America, like Venezuela, Colombia, to Caribbean. And those are diploid and tetraploid. So again, you need to look at the possibility of ploidy manipulation. Sweet potatoes are hexaploid. Wild species are diploid and tetraploid. How you can introduce a diploid characteristics into tetraploid? Can they be crossed directly? Little chance, yes, we can have the hexaploid diploid cross resulting triploid. But the triploid is dead to end. So then you have to manipulate it to make artificial hexaploid. So scheme is similar to potatoes, but case of sweet potato, genetic base information of wild species not so much. So less group of people working on. So other way around, there are a lot of opportunity you touch on sweet potato wild species. Okay. And this one is a typical example of the breeding in Japan. And actually, sweet potato breeding are quite common, and the major variety of sweet potatoes, even in Thailand. So you have the Japanese variety. So we have emphasis over sweet potato as the food security crop and the industrial application. So amazingly, they work with 50,000 seeds in Japanese sweet potato program every year. So the out of season, off season, people make a lot of profit to generate the project. But this is not sustainable in terms of how we don't have people and how much money you have to spend. So the next slide set, I will show you more genetic efficiency can be given by informatics. And actually, those over the NGS and informatics too to help the select more efficiently, select down to handle growth. But please look at the years to spend. Several years are still needed for the conventional scheme. So the crops, potatoes, sweet potatoes, you always need to have a long time frame and also G by E consideration to make the multi-year multi-location testing. And the background information, however, there is some the genetic tool coming out for your reference. Please look at that. There are a couple of groups also working on genomes, so different genotype of sweet potatoes, and deployed the sweet potato, the wild species, and they have a comparative genomic study on how they have evolution. And of course, genetic engineering option, because of the complexity of hexaploidy, relatively the smaller genetic variation, incorporation of artificial genetic variation could be option. But the problem of genetic engineering is low regeneration efficiency in the transformation and regeneration efficiency in transformation, including editing. So the marker application sweet potato. So sweet potato difficulty is the hexaploidy and the 6 alleles per locus. So even you go to NGS, how you make the the overlap with the 6 alleles. So there is need to have more informatics approach to sort out the different type of algorithm. And actually, yes, some work done recently by National Agricultural Resource Organization. And it's a reference that you can get also the open the download site. That I just would like to introduce how they made the hexaploid the genomic study analysis. So if you are interested in sweet potatoes, please look at this reference. So I did actually sweet potato also in the past, and I have my own review that make the complement on how sweet potato breeding can be done. And also it was almost 20 years ago, future look that today, how you can do sweet potato breeding. A little bit, maybe 5 minutes more, then I'd like those. Vegetatively propagated species, although nature or ploidy may be diploid, but you may handle the polyploid, the possibility to make an phenotype. So one example is orchid, consists of 15,000 species, like a dendrobium, phalaenopsis, already the triploid, tetraploid, the flowers made, and including Thailand, they are common. And rose, so there is wild rose, many places. But the common rose are tetraploid. Already it was artificially made as a large flower, attractive species. And the flowering industry, Ravera, Curcuma, Dianthus, Passiflora case, those are also fruit crop. So the polyploidization applied to make those urban. So the afternoon session, we will have how to make the polyploid out from diploid individuals. So that one is base for making some of the new varieties in horticulture. And the coaching, ordering are the common the entity, but those which also disturb the mitotic the chromosome behavior can be the breeder. So even the insecticide penicillin, which is the derivative out from the plant insecticide, could be the possibility. But usually it is recommended to use Orizarin, for example, or colchicine to make the somatic drug. This one would be the topics for afternoon, so I don't touch, and I just would like to introduce for. So finally, I'd like to close up with my present challenge with the Gibraltar in Southeast Asia. And the gingibrasi, you think about the ginger or turmeric or cardamom. And also you have nice flower like this as ornament, pedicure. So I think you have also similar one, Thailand. So because of global warming, even in Japan, we couldn't grow this over winter, but it persisted winter, so we could grow. So it is becoming a new flower variety summer, just this season. So there are a lot of potential to exploit the gingibrasi, and also it is a root rhizome crop. And also you look at the turmeric like substances, there is still a lot of potential to exploit the function and how you increase the dose over the compound. So there are different aspects of interest that we need to work. And the gingibrasi, the botanical future, I have some information, and even case of curcuma and also curcuma longa, which is a turmeric, and a ginger, gingiva officinalis, they have the genome sequencing. Case of ginger, there is a relatively large genome size, 1.5 gigabase. It's a large, I think, in terms of diploid, and the heterozygous is low. And there are the couple of major publications of NGS-based sequencing information, and the one of the inference of reason why, ginger flower, but not the fertile, could be the inversion in genome. So that one make the theory in ginger flower. But there is only short report made, so there need to have more exploitation. But looking at the generally like a turmeric and the ginger, there are some major targets, especially in interest of metabolite, like curcumin. The next slide set. How you can improve for the basic consideration for breeding approach. But the case of gingibraceae, generally speaking, expect some species flowering difficult, even faint flowers, sterile. So ordinary crossbreeding scheme is not easy. Then you need to look at those possibilities. So one. But before starting, you need to know how is characteristics of materials you are going to work on. And this one is an example of the Curcuma amada, Curcuma longa relative, which also produce curcuminoid. And the curcuminoid is the compounds the known to be effective for different the health enforcing functions. And we look at the different the accession that we could collect from, especially from Myanmar. And this one is a curcuminoid evaluation. These are the three different curcuminoid, total curcuminoid. And some shows the relatively high total curcuminoid and a different proportion of curcuminoid. And some don't have trace. And not all that so far we could look at the mitotic chromosome, but looking at the flow cytometry, there are tendency of polyploid having more components like this, especially highest one is polyploid. So the not moving to breeding, but evaluating the genetic resource in terms of how they are. And right now we need to do association study of a component amount and the ploid level. But there is the potential just look at the existing germplasm, evaluate, and find out the value like that. But this case, just an observation, but there could be possibility of the polyploid having a more compound effective for use. And indeed, a case of the curcuma longa, turmeric, there is the production of the polyploid, and that one would have the net the higher yield of curcuma. So we are looking at polyploid direction possibility on those of compounded yield in terms of selection for something that could anyway, Gingibraceae, based on what we have experienced on those of the polyploid association with metabolites, I have some summary of that we worked on, if you are interested. So I talked about 1 hour, and I thank you very much for your attention. And some of information stay in different publications. So please visit those publications and have your own insights. Thank you very much. So any question, ambiguity? I run many things. Yes, going back to conservativeness of plant breeding in any crops in history, not only potatoes. You look at the rice. The 30 years back, who think about the use of wild species? But now a lot of integration study made by one side the enough evaluation of wild species character, genetic marker available, genomics available. Now people moving to incorporate wild species. And potato, generally speaking, you go to Dr. Hans Ross, who is a German potato breeder. He started using tetraploid wild species maybe 50 years ago in his potato breeding breeding, history of breeding book. And starting, nobody agreed to use wild species because so much of the useless characters. And but the you point the resistance is availability not existing cultivated potato. So gap was big. The breeding, people want to have virus resistance, but you don't want to have large segregation of high glycoalkaloid, small plants, small yield because of virus character. So with that, some people still, like Dr. Hans Ross, continue, and he made the incorporation of virus resistance. So it was a 1960s or 70s starting, but the 1980s the bit of change occurred. So there are a lot of the wild species collection, German Dutch collection, and the USDA potato collection. At that time, also Russia had. So the gap was the field because of a scientific interest of reproductive biology study one way on incompatibilities, cross-compatibility, why haploid is made. So those baseline are very important part. And but again, beginning people didn't believe conservative potato breeders saw that it's the other another one. But in parallel, European bargaining continued. And gradually Germany, the Polish, they followed up. And also the United States, the group like pair I used to study at the Wisconsin, also the thought that the deploy in terms of simpler genetics and capturing the genetic diversity easily. But it didn't rocket started. Actually, we had to work on quite the genetic base to set up. So science doesn't catch up immediately with the reality. But with quite a tedious effort, maybe 2 generations, 3 generations science, we could. Is that your comment? Okay, thank you very much. It is a very good question. Generally speaking, gene editing, there are 3 categorizations in Japan. And one, like you mentioned, cisgenesis, especially knockouting and blocking this, some of the metabolic mechanism, they are out of a context of genetic engineering products. So we have the first example of tomato, GABA tomato, which enhance the higher amount of the health enhanced subject that is the outcome of genome editing. So depends on how technology judges. So that is a product-based evaluation, number one, in terms of authority. Number two, your question may go to the consumer's acceptance. And before commercializing this GABA tomato, seeds were supplied freely for users. 5,000 people are requested to test growing by themselves. So depends on how it is explained. Case of our case, not a high hurdle to provide a genome editing. Is that your question? they are principally allopolyploid with the disomic chromosome pairing. So the genetic behavior different like B or the BRCA type. And also, when actually you cross a cultivated tetraploid with those wild tetraploid, you can make a triploid. But the genetic imbalance takes place. So it's not only triploid per se. So the manipulation of a ploid is essential. So you have to put a hexaploid, a ploid down close to it. So it was also done by European potato breeders in the past. And especially the area of Phytophthora resistance, those coming with allo polyploid. So Europeans took a lot of time to make those incorporation of character. Yeah, endopolyploid can be common occurrence at a specific tissue on different species, as Dr. Hunt showed. So eventually it could take place on any place. And it also depends on how you handle manipulation, chemical treatment, it could cause the chimera. So but you want to stabilize. Like some of clonally propagated tree fruit crops, it could make chimera. So stabilize the genotype, you have to select a clone. So same for sweet potatoes actually. So depends on species how you say polyploid. It is endopolyploid, or it could be chimera. So by selecting a different the layer of a cell, you may squeeze out the polyploid. But endopolyploid is just a self-perpetuate on that specific species. There are a lot of classical examples. You go with all the cytogenetics book that how is a common occurrence. Yeah. Yeah, yeah. Endopolyploid. Yeah. Yes. Specifically, organs can be endopolyploid wizard. That's it, including the somatically doubled one, right?